I would like to introduce our speaker, Nari Talaty. And Nari comes from AbbVie. He's a principal research scientist at AbbVie. And today we're going to look at work that he's doing for MetID innovations in pharma, exploring new avenues for the metabolite structure elucidation. So in DMPK development for metabolite identification or MetID studies, they look at in vitro and in vivo systems, and this often produces heterogeneous mixtures for the metabolites. And with the analytical separation, the quantitation, and the accurate identification of these metabolite structures, this information is critical to the DMPK labs. And this is great work that can be done on a 7600 system for these ADME workflows. And today, Nari is here to give us a little bit of information on the workflows that he's doing. So, Nari, thank you for joining us today, and I will hand it over to you.
Thank you very much. I hope you can hear me, and I think my slides are up there. So thank you, Sykes, for the opportunity to speak. So I've been a Sykes user for a long time. You know, I've worked with their triple quads. I've worked with even the Q-Star Elite with a MALDI on it, and did some imaging work earlier. And now I have the pleasure working on the 7600 ZenoTOF. So I'm going to talk about some of the work we've been doing at AbbVie. This is, again, preliminary work. We have really started to explore the full potential of this system. We had a summer intern work with us at AbbVie. So I want to take a moment and acknowledge the co-authors here. Austin is in my team. Emily was the summer intern who worked with us. And then here are our folks. David Wagner is our manager of the drug metabolism and disposition group at AbbVie. So I'm going to jump right into it. What is human ADME and why do we care? So we take a medicine and we really want to understand what happens to it. So the fate of the drug, right? That is absolutely critical. We need to understand what's going on. So we want to look at the absorption. How does the drug get in? We want to look at circulating drug-related material. We want to identify and quantify this drug-related material. We want to understand where it distributes, how it metabolizes, which means the human metabolites that are formed. We want to understand all of these toxicological events, the drug-drug interaction liabilities, and ultimately, we want to understand where and how it leaves the body. And that's the elimination of the drug. What is the contribution of metabolism? And so our team, which is involved in doing all of these studies, provides a lot of information back. And we also submit results for new drug applications, the NDAs. And so our drug metabolism and disposition are the core area. We want to understand the safety and efficacy. We look, like I showed you on the previous slide, at the ADME, the data generated. So we influence from early in the pipeline all the way to like late stage, the human clinical trial. And so we want to influence the drug design and chemistry early on, and then later on look at all of these different things, which will then give clarity on what is happening to the drug. And our data goes and is part of regulatory filing. And we also provide scientific advice to several project teams along the way. So for discovery stage projects, we typically do and try to identify the metabolic hotspots. We look at the chemistry. We do early species, which is your sort of hepatocytes, S9s, the in vitro assays. And for development stage assays, we look for safety margins, like I said, and we look for the ADME data in human and preclinical species, right? So I'm going to go and lay this out in sort of a roadmap. So this is sort of our roadmap. So we have our tier 1 samples, and we look for the SAR, structure activity relationships. We try to identify some of the early hotspots, we look at liver S9. We look at those metabolites, the turnover in S9s. We look at exploratory in vivo MetID, where we will test urine in sort of animals. And so all of this is done in the sort of pre-candidate nomination stage. Once the candidate nomination happens, we have the candidate selection phase, and then we have the preclinical development. And this is where we do kind of the cross-species in vitro biotransformation studies. This is where this is required for our sort of IND filing. We try to select the correct species to do our sort of ADME studies. And rat ADME-like studies are actually done using a tritiated and a carbon-14 label. So this is where the radio labeling starts happening as well. Now, this is a busy slide, but this is also where all of our sort of clinical development MetID happens. So we do first in human studies. And in our group, we are really beginning to go with a sort of human first approach. We want to understand things that are important because ultimately the medicine is dosed in a human, and we want to understand what happens to it in a human. And then we want to compare and see that we have adequate coverage from our sort of other species, that is your rodent ADME as well as your non-rodent ADME, which are both carbon-14 label studies. These are also required for the NDA. And then you have your definitive human ADME study, which we want to start doing. There's also paradigm shifts in the human ADME study. We typically, traditionally look at carbon-14 labels, but we are also exploring other options where we can try to do these early, where we would have readouts by NMR. The mass spectrometer stays the same. So I'll kind of jump into that very quickly on what our tools are in a little bit. So here are some of the other assays we would support. So basically you can see it goes all the way from your phase 1 to phase 3 in the clinical trial. So now jumping into the drug metabolism. So all of our studies are typically our toolbox. If I were to put a picture of our toolbox, we have LC-MS, that is our main tool. We have our UPLC platform. We'll have different mass spectrometers, high resolution mass spectrometers. And the things we will measure, we have radioactivity counters as well, because we will be doing that. And we'll work with a lot of different tools and of the trade and do mass spec in the true traditional form, right? Because what we want to understand is what happens to the drug. So these are the different pathways of drug metabolism. You have your phase 1, which is your functionalization. So you have these different functional groups that would add to it. And I put everything in sort of the exact mass form, so everybody who does high-res can appreciate that. And then you have your phase 2 metabolism, which is sort of the conjugation that happens. And all of this happens so that the body can eliminate that drug, in whether it goes out in the urine or in the feces. So these are your different phase 2, your glucuronidation, your sulfation, phosphorylation, all of these are your phase 2. So you can see these, your phase 1 and your phase 2, and you're trying to measure that basically across the board for your ADME. So what we wanted to do was look at this sort of, do a comprehensive study of 40 different compounds, 5 belonging to each of these classes. So we identified the common sort of metabolites, right? Phase 1 and phase 2. We have your sort of O-glucuronide, your N-glucuronide, your acyl glucuronide. You have oxidation, the N-oxide, the sulfation, phosphorylation, and your glutathione conjugates, right? So you want to understand all of these sort of metabolites. And we wanted to do a thorough examination of the different techniques that are available to us. So we use different platforms. And we wanted to do a systematic evaluation of the different collision energy, like the different fragmentation tools. So here we go. So perform UVPD, EAD, HCD, and CID MS fragmentation to structurally elucidate 40 phase 1 and phase 2 metabolites across 8 different classes. That's what our task was for a summer intern this past couple of months. She just got done like 20 days back. So the different techniques are the CID and HCD. And then you have your sort of CID, which is your collision-induced dissociation, which is your classic MSMS, where you have nitrogen that would, or some sort of a gas that would collide with your ion of interest. It then fragments it. You have a higher energy collisional dissociation, which is associated more with your Orbi trap. And you have the way, ions are moved from your the C trap to the Orbi trap, and then you have a fragmentation that happens that is slightly higher energy, and you get a little bit more improved fragmentation. Your CID is classically your triple quad and your ion traps. HCD is in the Orbi traps. EAD is your electron activated dissociation, which is what we, is on our new 7600 ZenoTOF platform. And the fun that we really enjoy is that we get both the EAD and the CID on the same platform, and that's been really a joy. And then we also have a UVPD fitted onto one of our other platforms as well, which gives us another readout. And so UVPD is sort of, with your laser and your light and the duration of how long it takes, you expose it, and then you get a fragment. So I just want to jump right into some results, and by no means these are like complete. These are results that have just been generated. We've looked at them. We want to do a deeper dive into some of these things, but our first pass on all of these shows us, we've shown like CID and HCD basically, will just yield the simplest fragments. They're the collision happens. It's sort of it loses your conjugates. It doesn't give you a lot of structural information. The EAD actually retains the site of conjugation here, and that's what's shown by this asterisk. The 386 peak is actually retaining your phosphate. And so that gives you a little bit of better structural information, whereas all of these other techniques do not retain the phosphate. We want to jump into N-oxide and N-oxide. And again, we start seeing that the EAD is useful. It kind of retains part of the N-oxide. The UVPD, HCD, and CID, you just get straight up collision that doesn't. This is a lot of work is still going on. We're really getting to the bottom of these. We are looking at these spectra more and more. We are using different ways to process this data, but every which way we look at it, we're getting richer fragmentation with our EAD than any other technique at this point. And the EAD has been especially useful almost immediately off the, like as soon as we started running it, for identifying our sort of glucuronides. And in the glucuronides, if you look at the CID and HCD, all that happens is that you have a straight loss of the glucuronide and not many useful fragments that actually retain the gluc. And so having a structure, having a fragment like shown here, which is the M/Z329 that actually retains the gluc, this is marked right here, is very, very useful. And as we dig more and more, so one of the things we learned, and I do want to pass this on as, for people who begin to use the SciEx and begin to use the 7600 and EAD in particular, is that you have to overcome a mindset. If you are used to looking at HCD and CID data, right, you're used to these sort of really high intensity peaks. And with our EAD, it fragments differently. The mechanism, the mode, everything is different. And so there is a little bit of a difference in your mindset. You have to be prepared to look at the sort of low intensity fragments. You have to be able to explore that. You have to be able to understand that, and you have to be able to accept that. And now the fact that we're working with high resolution instruments, we're working with low ppm accuracy on each of our fragments, we can comfortably identify them. So this is something that all of us in our group had to sort of, okay, we are looking at fragments that genuinely have lower intensity than we are used to seeing with, let's say, HCD or CID. UVPD and EAD are sort of in the similar realm, but EAD is giving us definitely a richer set of fragments at this point. For the compounds that we've tested, if there is double bonds, the UVPD tends to have a little bit of an edge. So I think all of these techniques eventually you'll see are complementary. to each other as well. So again, moving on to another N-gluc. Again, we are retaining part of the glucuronide. We have some fragments that actually retain the gluc. But if you look at the CID and HCD, again, it's like a nice little smooth, there's nothing here, but our EAD begins to give us some fragments. There's no fragments here with the UVPD either. So this is something that is really useful to us because we, in the development MetID space, our entire goal is to not miss anything. And we want to do comprehensive MetID. We want to go deep into the structure. We want to understand and solve the structure as much as possible. Eventually, we would have to give some of them to NMR, but a tool like EAD allows us to get good data early. Now, I do want to point out that not everything works, so it's not all rosy, and I'm only showing you a select few examples here. And I'll kind of summarize at the end how many of these 40 actually worked and what we did with them. But some of these, like the sulfates, the negative ion mode doesn't work. We consulted with SciEx. We bumped up the collision energy to almost 20 here and 25, and we still don't see, and it just starts falling apart. So some of these things do not work, and that's pretty normal with any technique. Now, I want to jump a little bit into what our development studies and workflows look like. And I just want to give a brief overview. I cannot show details on actual studies, so I'm just going to give arbitrary numbers for metabolites. So this is something where we do, this is from one of our rat ADME studies, and this is a radio profile of a rat bile. So we have a really long gradient, and we try to identify as many of these peaks as we can. And you can see there's a whole bunch of metabolites that are labeled here all the way, we find quite a few. So the ones that are annotated and marked here are all major metabolites. That means they're above a sort of 10% threshold. And those are the ones that we're really interested in. And the samples are all acquired by sort of the LCMS, and we fraction collect, and we get a radio profile. So you'll get your LCMS readout, and you get your radio on the same sample. And then we ran this on our SciEx. So this was trying to benchmark our Zeno with our existing instrumentation in the lab, our other high resolution platform. And we want to, I want to give you this, that having a radio is fun, is really, really fun for people who come. I came into the development sort of MetID group from a discovery side. And on the discovery side, universal detector was our charged aerosol detector. And here the universal detector is sort of the radio. The mass spec suffers from its sort of ionization efficiency, right? I mean, that's something we all have to consider. Not everything ionizes the same. But radio, it's just constant. It gives you perfect counts. So basically, the radio gives you an unbiased profile, and then looking at the mass spec, we were able to see that the XIC of all of our major metabolites they all showed up on the SciEx, and they were very, very comparable with our thermo. So this was our first sort of foray into doing one of our MetIDs on a SciEx instrument, and it really showed us some very, very comparable and good data. Now, the thing that drew us to the SciEx was that you not only can collect data in different forms, you can post-process the data in different forms. The software is powerful. It allows you to do many things at the back end of your workflow. So you have your isotope pattern chromatogram. You can actually collect data in that mode, or you can process the data in that mode. And doing this sort of isotope pattern chromatogram will get rid of your sort of false positives. And so this is why having something like this to filter your data. So here again, the XIC of major metabolites is shown. This is from the ZenoTOF. This is the pre-dub data, but this is something that is straight off the instrument and hot off the press. We're beginning to use this in our workflow, and it actually, the data begins to look good. It gives you a very good idea of what is being picked up and what's real and what's not. So this is one way to actually not just process, but also filter. So this is post-process, so it's post-acquisition. Again, we use mass defect filter. We may have to modify this a little bit, but again, we can look at the different peaks of interest, and it tells us which of these peaks are real, which of these peaks are not. we need to incorporate this into our data acquisition. This is something our director from our team has always, he's been doing this for 20, 30 years, and he kind of told me this is something that is really powerful and we need to use this. So we are beginning to scratch the surface. We are beginning to use this into our acquisition workflows now. We have always used this in the sort of post acquisition. So I'm not showing that data yet, but we are using sort of three different, it's really powerful to have three ways to acquire the data, three ways to process the data. It really makes it difficult to miss things. Now, our discovery workflows, actually, what do we do, right? What is the 7600 going to do for us? So we really want to use the speed of the TOF, and the TOF goes very fast for acquisition. So we really, in terms of your other, Orbitrap or any other analyzer, the TOF will have the advantage of speed. And in discovery, we always want to do our assays faster, quicker. We want to get everything out back to the chemist quickly so they can make a decision. They can change if they want to change something on the fly. We want to automate our data acquisition workflow. And so we are working with our SciEx colleagues. They're helping us to do that. Again, DDA, mass defect filter, and isotope pattern. I actually mentioned that on the previous slide. So those are the ones we will use, and we will do quick and agile studies. That's the ultimate goal. We want to do most of our in vitro work using this platform. We want to industrialize the 7600. We want it to be like a workhorse. Now, the other thing we want to do with it is we have two platforms. We have two 7600s. One will be dedicated to our discovery work. One will be dedicated to our development workflow. Now, in the development side, like I said, we cannot miss anything. We want to take every molecule, basically, that we identify, every metabolite, and we want to understand every detail about it. So that's where, again, having the sort of three different modes, doing the different fragmentations, it really, really helps us. We start with our cross species, where we do that. Then we do our sort of ADME studies, and then we do ultimately all of our human ADME studies as well. So there's a lot of this data processing that we want to do. So this is our sort of workflow, which will be going into, so we also want to do backend a lot of work on software modification, right? So we want to take our molecular profiler and we want to take mass metasite as well as our custom home-built software. And we want to do a lot of different things with it. We are working very closely with our colleagues in SciEx to do a lot of different assays using this software platforms. So I just want to jump in and throw a different curveball here and talk about a completely different technique. So we also, as a classical mass spectrometry group, we also have an FTICR, which was actually purchased for imaging. And we are now using one of the sort of the SORI mode, which is the sustained off resonance CID for the FTICR, to see if that offers any inherent advantages. And once again, yes, you do get some of the peaks, but you have to remember, I'm going back here, this is not coupled to any LCMS. So this is a sort of standalone flow injection platform. So it's not a very practical way to do things, but it will give you some information. Once again, this data compares with EAD. So if you can obtain similar results, again, the EAD is sort of doing and standing on its own, even when compared with this sort of FTICR SORI platform. So I want to take a minute on this slide because I do want to get my point across here. So we have performed CID, HCD, UVPD, EAD, and SORI on 40 different metabolites from 8 different classes. We optimized different fragmentation energies. So for each one, we took, did a range. We did a lot of work trying to understand which of these. This generated a lot of files for our summer intern. She did a great job here. The conclusion was 25 of the 40 metabolites, EAD, yielded unique fragment ions, and when compared to the CID. And the beauty was that you have EAD and CID in your same platform. And I think you've seen this from some previous presentations as well, that is the power, that you're able to do these two things simultaneously we are really going deeper by using different software, including our home-built software, which one of the members, one of our co-authors here, Rich, has developed a home-built sort of software that goes deep into doing MetID. We're trying to use that. We're trying to see if that can couple with some of the stuff we have. We can feed into the SciEx profiler software and see if we can get some readouts. So that's another thing we're working on with Ava from SciEx. Rahul and Ava are really helping us with some of these things. EAD is applied for human and animal ADME studies. It's become a part of our workflow. We see anything interesting, we throw it on our 7600 and we try to generate sort of the EAD fragmentation workflows for that, fragmentation patterns for that to get more. Again, the overall conclusion that EAD and CID are complementary, and when they combine, they will cover most of your needed fragments. And again, we're working hard to build up this decision tree because every presentation I give, it's like, what is your conclusion? Why have you not yet come up that this works for this doesn't work for this? It's not a one size fits all. There are some conclusions that, yes, glucuronides work beautifully, and SciEx has published on this extensively, and there have been some great presentations at ASMS as well. We're not the first to do this, but we're definitely finding it useful. We're seeing these peaks, we're seeing them, hey, it's like discovering these extra peaks that totally make sense and then allow you to solve the structure. So definitely the EAD is powerful. We are kind of finding it so far. We're struggling with the negative. We want to get more involved. So that's where someone like Rahul, Randy, Shaw, they've really been helping us. Ava has been helping us, and Todd and Spencer have been helping us with a lot of the logistics of how to get things done and get the most out of our two systems that are at AbbVie right now. So I'm happy to take any questions you may have. And thank you.